And it's my pleasure to actually talk about it, facilitating eradication of polio virus and other vaccine-preventable diseases. And these include like things such as measles, mumps, rubella, and so forth. This is part of this history of medical science, and it's a pleasure of mine to actually give you this talk. And what I'd like to talk about today is the history of polio virus, and then the polio vaccines that are used today and how they're changing, the face is changing as we further eradicate the virus, our challenges in vaccine and biotherapeutic manufacturing. So this is an important point. So it's the way that we have changed forever the way vaccines are produced. And I'll describe that at the end of the talk and tell you how the solution value proposition has led to formation of a company here at UGA that has over 12 subcontracts with largest biomanufacturers in vaccines in the world. And all of this is all attributed to the Gates Foundation. I want to bring that up now, tell you that they funded this from the start. They're still funding it now. So for those that don't know the history, and surprisingly, most people think there's no more polio because it's gone. And in fact, that's far from the case, but I'll tell you as we go. So polio in the 20th century was one of the most feared diseases of all because you had tens of thousands of kids paralyzed across the world. And of people being infected with polio, only 1 to 2% actually show symptoms. So that tells you how widespread people were infected. So soon after the introduction of vaccines, OPV and IPV in the 50s and 60s, polio was brought under control, but not before it scared everybody on both sides of the Atlantic. Because when the epidemic happened, this kind of thing happened. Pools were shut down, towns were closed, you couldn't go to movie theaters, period. And that was only about 30, 40 years ago. So many of you probably were very familiar that are a little bit older. Others think that polio is gone. Polio is a common name for poliomyelitis, and this was a common occurrence back, it still is today actually, but more common back about 20 years ago. A very infectious disease caused by contamination of feces and water supply. And virus replicated in your gastrointestinal tract, where it leached into your blood, provide level of immunity. And about 1 to 2% of kids, you would get some form of poliomyelitis. Polio itself is called a enterovirus. It's from the genus. There's, like I noted, about 95% of all cases never have anything associated with them. So you don't even know if you have them. You could have asymptomatic polio, or border polio, where you have maybe a fever, you feel a little bit achy, and you think, oh, it's the flu. Even today, this happens all the time. Then there's non-paralytic polio, where you add the symptoms of this with now neck pain, you might have muscle pain, some level of neuronal issue. And then in the 1% or 2% of people, you have these loss of muscle reflexes where people become paralyzed. And so here's the king doing his thing. And this actually was representative of the hospitals back just a little time ago, and still is in some countries today. Kids and primarily children were stuck in these hospitals in these iron lungs. And they were lived there a whole life until they had to either take a pee or change their clothes, or they were allowed out to eat, and that was it. They went right back in. Because otherwise, you couldn't breathe, and they would die. So iron lungs is not that long ago. This is something that's a part of history in one sense, but realistic and what could happen should vaccine fails. And we'll talk a bit about this. So as you look at poliovirus in a microscope, this is an RNA virus. Essentially, it's broken into 3 types based upon the capsid, which is the protein complex surrounding the virus. So the capsids are type 1, type 2, and 3. We have polio type 1, type 2, and 3. Type 2 poliovirus is eliminated, no longer exists. It was in India back in the late 80s. We have not seen another case since. Type 3 is almost gone, but still circulating in Nigeria, Afghanistan, and some other countries. And now, really, the type 1 polio is the most pervasive strain of all. It's a bugger. It's actually in developed countries. It was recently in Israel. It's a pretty developed country. And cases of it occurred off in South America, which are pretty developed as well. Polio can be transmitted basically by contact, as I noted. It's a contact, hand-to-mouth, fomite, generally. It also is done by the water that's contaminated. A lot of people, this is the source system in Israel where people contracted polio. I think it was last year or year before. This is open air, so clearly there's aerosolized crap. People contracted it through this, about 131 people. Then there's practices, religious and weddings and things that happen in water. And water in most countries is pretty bad. And finally, you have these watering holes, if you will, where people gather. And not everyone is conscientious of their brother or friend. This water gets pooed in quite regularly. And so any of these ways is typical transmission of polio. So polio enters through the mouth. It generally grows in the respiratory tract, and then it evades from there. It can evade and cause everything from, everything from subtle poliomyelitis to more severe forms. I want to give you a history about polio because this is quite interesting and relevant to the lecture series. So there's about 6 slides of timeline, and I'm going to talk a bit about this. So as far as records go, back in the 1400 BC, this is an Egyptian slab that went over a tomb. There was cases and carvings and hieroglyphs, whatever, that had indicated that these pharaohs had the phenotype of polio, this weakened leg, and this is a crutch. And the bottom line is this pharaoh probably died from polio. But in 1894, however, the first outbreak of infantile paralysis that actually was attributable to polio occurred, and this kind of scared most people in the United States because this was our first endemic, really, of this. In 1908, a lot of things happened between that and 1908, but the Australian physicians Carl Landsteiner and Dr. Popper discovered that, in fact, the cause of AIDS might be a virus. Polio had never been, it was one of these things they thought bad fumes, all this stuff, this weird stuff in the old days. But in fact, they had evidence from a electron micrograph that this was probably a virus. Then in 1916, the first polio epidemic hit New York. New York was still is a major city, but relative to other cities back then, it was huge. This scared everybody because this is what was resulted from that epidemic. All these kids, look at that, these are infants. They were stuck in these iron lungs. And later, of course, their lives were shortened by polio. In 1938, the United States wanted to do something about it. So they established a infantile paralysis fund, if you will, later turned into the March of Dimes. And so for me, I remember when I was a kid on Halloween, we'd have trick or treat for UNICEF, trick or treat for March of Dimes. We were trying to raise money by going to doors, collecting candy, and getting dimes or quarters for the fight against polio. And I'm not that old, but this is how recent this actually occurred. So this is within our lifetime. By 1955, we had our first real vaccine. This was done by Jonas Salk. It was an inactivated polio virus, or IPV. And this was really the best virus we had at the time because this kill vaccine would induce immunity, but you could not get reinfected through the vaccine. Because I'm going to tell you, as we go talk about today, most of the cases of polio that happen today are from actually vaccine-derived polio. So we use OPV, oral polio vaccine, which was developed in '61 by Sabin. This live vaccine contains a weakened virus. So you get it, you get mildly infected, if you're healthy, you clear it, and essentially you have no symptoms from it. But remember where this vaccine's going. It's for third world countries primarily, and they are far from healthy. They don't have proper diets, they don't have proper anything. So that ultimately is where the source of polio is from today. And I'll talk about this in a moment as well. In '74, the WHO World Health Assembly passed a resolution to try and target polio using an expanded program of immunization, or EPI, because back then we were just doing pockets. If someone had a polio case in Kansas, they'd send people out there and try and do herd immunity, where you immunize people within the town to protect the rest. But that actually never worked. Polio is so widespread, so easily infectable. From the 70s and 80s, the CDC and others collected surveys from around the world and said, are any of your people lame? Do they have problems walking? Do they have issues that would be associated with polio? And the answer was a resounding yes, so they introduced routine immunization with OPV to many different countries. course, immunization at that level costs money. And so this is days before Gates and Bill and Melinda Gates Foundation. The Rotary Club is a huge, huge supporter of polio immunization. You guys should support them. They are private sector, support this initiative, and they raised initially $120 million. Doesn't sound like a lot, but from zero, that is huge. And by 1988, the World Health Assembly, or WHO, passed a resolution, 121 countries, that said, okay, we're buying in. We're going to donate money, and we're going to try and get rid of polio. Because in red, this is how the world looked back then for wild-type polio. And that was bad. Then in 1991, we had the last case of wild polio occur in the Americas. And this occurred in this child. He was the last of the last in North and South America. He's Luis Tenorio. He was living in Peru, northern Peru. And because of continued vaccination in those countries, he's the last one in the Americas. To be declared polio-free, you must go 3 years without a polio case. And this happened from 91 to 94. And WHO declared the Americas were certified polio-free. So that's a huge step. That's actually where it all started. And then in China, because they saw that happen here, they've been vaccinating children by the millions. They vaccinated over 80 million during this 1 year alone. You guys probably heard Nelson Mandela. He's very important in this campaign. In 1996, he had kicked polio out of Africa campaign. Because Africa, the one thing that unites them is soccer or football, if you're somewhat not U.S. But in the whole business, they raised enough money to vaccinate 420 million children. And so that was a big step forward because Africa, they rather vaccinate against things that kill them. Things like HIV, stuff like this. To vaccinate against polio, which only inflicts 1 to 2%, doesn't make economic sense to them. But this started their whole thing. We got to get rid of this thing in Africa. In 1997, this is Mum Chanty. She's the last case of wild-type polio in the Pacific region. So she was from Cambodia, I think Phnom Penh, but bottom line is she was the last one. And then things started to come together around the world. We started seeing more last cases. This occurred in Europe. This person was Malik Minus, a 33-year-old, month-old unvaccinated child from Turkey, and the last person from Europe. And then in 1999, we thought everything was going really good. But because of essentially war that occurred in Liberia and some other locations, they stopped vaccinating. And so for that 1 year they stopped, we had a huge outbreak of polio in that area. Here's a fellow carrying live attenuated oral polio or Sabin vaccine. The UN Secretary General negotiated a truce for I think it was a day or two. They vaccinated millions of people in the DRC, and basically is an example of that. Then in 2000, we had the Western Pacific region declared polio-free. And so more than 1/10 of the world's population received OPV during this year. So that's a crap load of kids. That's 550 million at one shot. We thought it was a big deal. But then in 2001, the next year, we did 575 million. And this was through people like the Rotary Club and so forth. They got off their butt and went to Africa and helped vaccinate because that's the only way you can get it done. And in fact, we have more than 35 million in Afghanistan and Pakistan, which is still polio positive. Still wild-type polio circulates there today. 2002, we have declaring of the European region free. And so this was 500 million kids were vaccinated. 2003, we have Nigeria. There's more unfounded rumors. A lot of people, this is a problem with polio, with measles, all these other vaccines. You take it outside the U.S., a lot of people think we're bringing in something to exterminate them. And so unfounded rumors said that the polio vaccines are going to kill millions of kids. So all the moms and dads stopped vaccinating their kids. It didn't even take a year, another outbreak occurred. Because the second you drop your guard, polio is on you. And so when we started revaccinating and brought polio back to control, 2004, we had synchronized National Immunization Day. And this is the way that it's done now. But we are immunizing 80 million children. That's a lot of kids, all on the same day with the same vaccine. And this is the oral polio as shown here. In 2005, we developed and approved a monovalent vaccine. This is important because the vaccine kids have been getting up until today was a trivalent with OPV 1, 2, and 3. And OPV 2 is exterminated, so it's not here. So no reason to put it in the vaccine because with OPV, you get release of the weakened virus that maintains the population of OPV 2. So we had supplemental vaccine here, and this is really important because when there's an outbreak somewhere, it's better to immunize with the kids with the virus for which the outbreak started than to give them this trivalent thing when you do OPV. Everything's OPV, and I'll discuss this in a minute because you only needed one vaccination. Because the vaccine would slightly grow in you, you would recover from showing any disease. It was very potent and effective. But it turned out now that OPV absolutely has to be replaced if you want to get rid of polio because the Sabin vaccine's got the weakened strain and Salk has got the killed. And the killed will give you the immunity, but it costs 10 times the amount of OPV. And so I'm going to describe at the end why our technology we have is needed to facilitate the eradication of polio. So as you've seen, in 1988, we had 120 plus countries. We had polio out just about everywhere. By 2006, there was 4 countries that had endemic wild-type polio, and it wasn't even hot. The countries had it, and because they had it, they could spread it to China, they could spread it to the U.S., where people travel. They go on boats, planes, automobiles. So we started with a more closed-in timeline. We thought that we can control outbreaks now. We thought the vaccines would be good, but we learned that, of course, polio eradication comes at a cost. A lot of dollars, a lot of effort, a lot of time. You got to find that 1% of the people that are distributed in these back countries and vaccinate them, because wherever they are, polio will find them. And this happened with Nigeria. So all these light blue countries came after we thought we had polio controlled because we had wild-type polio here and just spread around. And so now we're looking at 2016 as a real timeline. Where India, for an example, has been declared polio-free. It's gone 3 years without a case, which is amazing because there's millions, if not billion, people there. And that shows that it can be done because that's not a developed country in many ways. And so I think it's just now making sure we address these issues. So we're beating back the devil. Today, there's 70 wild-type cases worldwide, and about 26% of these cases are caused by OPV. OPV, the weakened form, mutates in the intestines, and it becomes wild-type. This was compared to what we saw last year, which was 341 wild-type cases and 51 vaccine-derived. So it looks like the noose is tightening on polio, which is great. We need everyone's participation. We can't just go at it and say, oh, this isn't a big deal. Literally, if we stop vaccinating in places where the stuff is near or circulating, you'll get it right back. Today, there's 3 countries that have polio. They have not been able to stop it, even with vaccine campaigns. So we have these kids, essentially there's children are the most susceptible around age 5. And this was a poll taken, I guess, in 2011. And so it was asked then by the Rotary Club, asking the people that are members, tell us about polio. And 44% couldn't even believe it even existed. They said, oh, it's a disease of the past. In fact, they're voting all their money in the effort, 122 million Rotarians going after these kids. And the cost back then was 60 cents a dose for oral polio. Inactivated polio was $6 per vaccine. And if you're in a third world country, you can't afford even 60 cents, let alone $6. So what I'm going to tell you today is how we've changed these numbers, how we've developed enhanced vaccine cell lines so we can make vaccine at 15 cents a dose. So it can be globally distributed and we can eradicate polio, measles, and other things where the virus only circulates in humans. And as I mentioned, to declare disease eradication, you have to go 3 years without the disease. And so since there's no new case, well, there's a few new cases potentially this year. Most of it's vaccine derived. But it looks like we might actually achieve our mission by 2018. Why I want to bring up the vaccine-derived polio is that's the only source of polio around the world today. There are millions of people vaccinated with OPV, the weakened form of polio. It's in their gut circulating now. In about 1 in 200 people, the virus mutates, and as shown here, you get these mutations occur, and this changes polio. And the polio will come out hot and nasty, and sometimes it avoids the vaccine, which is really bad. So we have mop-up campaigns we go after polio with, and our group is associated with a lot of work the CDC and WHO does. But this vaccine-associated paralytic polio is still occurring. About, in this case, about 2 million kids get it around the world today. So we're at a point where we need to switch entirely our vaccine approach to IPV, or the inactivated killed form. Problem there is this, like I said, is $10 a dose. Getting IPV is really up in the ante and the burden on the economics of the kids. But we needed to induce the risks of polio, as we're withdrawing OPV2 now, oral polio vaccine, so we need to replace it with the killed one. We need to be able to interrupt transmission, so you supplemental vaccination. When a war starts in Libya or Afghanistan, these things are, people are always fighting there. We need to be able to have a truce day, fly in, no one gets killed or captured, vaccinate the kids and fly out. This is still happening today. People are being captured because they're trying to save lives, is the bottom line. And ISIS, if you love them or not, they're the ones that are causing all these problems right now. And so, you know, it's hard to tell from people. People have different standards of life. And most people think vaccination is a good thing. But ISIS has a belief that we're there to do them harm, modify the way they live, sneak up on them and stuff. And I have to be honest with you, I don't know if you know this, but the CIA actually had a campaign. They initiated a false vaccine campaign so they can go in and try and get bin Laden and some of their people's blood and genetic material. I kid you not. And because they did that, no one trusts us now, where we're going. It was the lamest thing. It should totally have been allowed. I would have personally taken all those guys and thrown them in jail. But they're out there today. So this is part of the problem. And then we want to be able to hasten eradication. Because we're really close. We're this close to getting it done. So inactivated polio right now contains 3 serotypes. And we also can do 1 serotype at a time. Finally got approval. But they grow these in viral cells, which are monkey kidney cells. And then this is inactivated with formaldehyde. And so it contains a bunch of things, the things that are mainly preservatives, like polymyxin Bs for fungus and so forth. So there's nothing special about this vaccine other than it's a killed form of polio. And this is sort of the schedule that they were on. It used to be IPV, OPV, IPV. Now it's IPV all the way. So OPV is being ruled out. And this is what's been agreed upon with the legacy plan. So the legacy plan, the objectives are shown here, what they want to do across the time from 2013 to 2019. We're right here now, the last use of OPV2. We're going into totally complete IPV vaccination worldwide. Everybody is vaccinated now with IPV. The problem, like I said, is it doesn't have the same efficacy as OPV. You have to give more than 1 dose to protect 95% of the people. But we have to do it because it's the only way we can eradicate. Pardon me. So what I want to do now is talk about our polio program here at UGA. And with our company, we've started based upon some platform enabling technology we're sharing with Stefan's team and the people at GRA, Georgia Research Alliance. So the goal is to eradicate polio. And I told you a lot about vaccination and history, but today, 2 billion people are vaccinated each year for polio. And someone's got to pay for all that. So what do? You come up with the best way to lower the cost. 150 million kids are vaccinated one day in India. This is what it takes to control polio. It takes your friends, your neighbors to get up and try and invest in people, private sector people that do this. And you can see them all carrying these coolers. These coolers contain oral polio vaccine. That was from 2 years ago. Now it's inactivated polio, but they still keep it cold. So So what's hindering eradication today? Well, eradication is being hindered by the cost of vaccine manufacture. Money's everything. That's what it is. And we also have an insufficient supply, so we can't keep making this and storing it, letting it decay and being not useful. If you talk to BARDA, they don't care about storing polio vaccine. That's not, we eradicated it here. These other countries don't store vaccine. Someone has to store it. They have to put in high titered vaccine that will decay over time because it naturally occurs, but it's still useful for stamping out outbreaks. So we have limited supplementary immunization ability, and we need a switch, which to a vaccine that's 10 times, it's actually over 10 times the cost. So what's your solution? Your solution, of course, is to create sufficient amounts of vaccine at affordable prices. What I'm going to tell you today about how we discover which host genes in the vaccine cell lines restrict the growth of polio and all its serotypes, how we can knock those genes out and create stable vaccine cell lines, and overcome the cost and the load of getting the polio vaccine out to the world, providing global access. So the underlying causes of all these are the things you might suspect, everything from difficulty growing the virus, because things happen, to increased global demand, to supplementary immunization. When there's a war, they say, oh, we're going to let you go in tomorrow to vaccinate. You have to have the vaccine ready. You can't just, oh, give me 2 weeks. No, you got tomorrow to do it. And then shit happens again. So we do what we can. The solution and approach here is we go after taking the cell lines. So remember, the vaccine cell lines for polio are vero cells, the monkey kidney cells. So what I'm going to tell you about is how we use an approach that allows us to screen the whole genome of monkey kidney cells to determine which genes are needed or manipulate the virus. Identify and validate those genes and then engineer the cell line so we can go from one virus particle to greater than 88 just by growing it in these enhanced cell lines. And so the benefits are pretty clear. I guess you probably know we can have increased capacity, reduce production costs, make the 15-cent vaccine, which is needed. And then this manufacturing footprint is big because if you talk with a vaccine manufacturer, they grow other things in polio. They're growing flu, they're growing measles, they're growing hepatitis, whatever vaccine it may be. You got to give them rationale why they have to stop what they're doing to grow a batch of polio vaccine. And that's basically money. If they don't make money, they don't do it. These people are not your friends. They're all about money. So we can reduce this manufacturing footprint. If I told you I could grow 80-fold more vaccine, instead of having 80 biomanufacturing units or fermenters in place, you need one. It allows you to do all this other stuff, and for them, then that's a reasonable deal. And so right now, we're able to achieve our goals very rapidly. Pardon me. So the technology of how it works is roughly shown here. We look at every single gene in the genome, every single one. And we ask, does that gene participate in the growth of this virus or hinder the growth? Because the bottom line is a virus is an obligate parasite. That's what it is. It grows in your cells. It has to use the gene products of your cell to grow. And the cells have inherent mechanisms that inhibit viruses naturally. So we find both ends. We find those genes that help the virus, those genes that inhibit the virus. And then once we do that, we go through a validation process using RNAi, small interfering RNAs, which is a transient way to knock down gene expression. Typically, it knocks down 85% expression. When we finally confirm and validate, we go to the next step, which is to permanently knock down the gene in a stable cell line. Because we want to get a stable cell line down to a manufacturer. It could be Novartis, it could be anybody, and it's theirs, and it's used to grow polio. And so the bottom line, we generate these permanent knockouts through a process called CRISPR. And so I'll talk briefly about that. So the way that we actually go through the process of analyzing the genes in the screen is we have a pool of siRNAs, and the siRNAs target the genes, recognize them, they knock them down. They essentially clip the messenger RNA. We transfect these genes, these siRNAs, into a Hep2 cell line. Hep2 is a human cell line. The reason why we're doing that is because most of the vaccine cell lines from viral cells to MRC5 or NDCKs, which are used today, the genome, full genome, is not known. So we have the burden of sequencing the genome of the cell line, and to first know which genes and to target, and then we have to design the sRNAs to target those genes. Then we screen, then we validate. So it's very difficult. Here we started just with Hep2 because we didn't have the genome for viral cells, monkey kidney. And we then infected with the Sabin2 virus and looked at the antigen that was released. And so this matters for the vaccine. The vaccine, all they care about is the antigen. Is the vaccine going to induce immunity or not? After the analysis, you generate a Z-score. So these genes are 3 standard deviations from the mean. So they're more likely to be positive. What we do is we find those genes that are here, and those are genes that are likely enhance virus replication. For us, it's important to knock those out to make the vaccine cell lines. So they're restricting the growth of polio. So that's what we do. We also have double-edged sword. We also find the genes here that are identified druggable targets. Those are host genes that the virus absolutely needs to grow. So we can actually identify those genes and create drugs to knock those out. So you want to have both ends, because the only way we can stop polio is to make sure that we get enough vaccination through an activated vaccine, but then mop it up with the antivirals. Because you got to remember, every person that's got polio in the world ranges from 5 years old to 50 years old. Polio transmission stops when those people die, and the reservoir goes away, right? So the trick is you either let them die, or you come after them with an antiviral and remove the polio, OPV polio they have. So we're addressing both things. We identify multiple knockdown events that actually enhance the poliovirus replication. Once we get our list of hits, we then got to validate them. And what we do is take the pool of sRNAs, so originally there's 4 against every single gene in the genome. We break them down to individual genes and ask which of those sRNAs is doing the work, or is it multiple genes, multiple sRNAs. Then we actually test our hits in viral cells, because the objective is not to change the way you manufacture vaccines. If you do that, you're going to set yourself back 17, 20 years. We'll never get to the end. We want to be able to plug into the system now, even though we know there's better ways to do it. We have to use the technology they have, where we just change the substrate used to make the vaccine. We take their viral cells, knock the genes out, and hand them back to them. And then we will need to test the antigenicity, because if you're making virus in the vaccine cell line that's not equivalent, you might as well start over, throw the whole thing away. So we want to make sure that the knockdowns don't alter the antigenicity. They're making enough neutralizing antibodies, etc. Then you have to test them against all the strains of polio that are known. You just can't pick one and say willy-nilly, oh, it works for this, but not that. It's no good. That's not good enough to eradicate the virus. So you ensure the genes work with all the vaccine viruses, and then you prioritize. And this is really based on translation industry. There's a variety of people we work with today, from the Serum Institute of India to Merck. And those people all have their own priority about what's going to happen next. So key is eliminating false positives. You don't want to pick the wrong gene and go through the whole process. So we deconvolute the pool to individual sRNAs, the singles. We test them again in Hep2 cells, and we find that when we tested them after doing this, we lost 50% of our hits. So 50% of what looked to be good in the screen was off target. It didn't matter. So we found the right hits, and that we then went down and reduced those and found 60 that worked for all the strains. Then we moved from the Hep2 cells, which is the human cell line, because we didn't have the viral genome, and tested in viral cells. So we're repeating the whole process again. Here's the viral cells. We take Sabin2, or one of the viruses, individual sRNAs, and we look at 2 endpoint assays to ensure that what we find is right. And so the thing here, this is a plaque assay. Each one of these white spots represents a clone of virus. So what we do is, this is what you get back using the standard viral cell lines for polio. And it's not that much virus, but yeah, that's how it's done. When you knock the genes out, we see huge increases from here. There's an example from here to here, here to here, etc. So we'll find single gene events we can knock down and get these large increases. In general, we find events that will lead to greater than 20-fold. Some cases it's 80-fold, some cases about 25-fold. But in general, If we achieved a twofold increase, or definitely threefold, Gates would have been happy at that. Because that means we can make enough vaccine for global access to cover the world and still lower the cost. But now we're up to expectations that are 5 or 10 times what Gates had. We needed next to see whether this worked on all polio strains, because when you're making the vaccine, you don't make it just for one. These are multivalent vaccines. So here again, now we're working viral cells because we know what hits to use in there. And we're looking at all the different viruses that are polio strains. And bottom line is we do plaque assays where we measure quantitate the amount of virus we get back. And so this is the control. You can see low amounts of polio. But when you knock the genes out, a single knockout of one gene that restricts polio replication, you get huge increases in the amount of polio. So again, we can find the hits from Sabin2 that we did our screen on can be applied to all strains of polio, and that's really important. Next steps are antigenicity. I told you what good is this whole technology if we're not inducing a proper immune response. That's going to protect the kids or 5-year-olds. Again, we're looking here in viral cells. We do the genes are transfected in the cells. We knock them down, generate virus, and we ask, does this human serum from already exposed patients, this is serum from people that had polio or have it, or vaccinated with polio, does it recognize the virus we're generating in these enhanced cell lines? And the answer is a resounding yes. It's exactly the same. What we thought would happen, but you have to test it. And so we end up finding that looking at 13 targets, we knocked out 13 different genes, knocked them down. We tried a bunch of different viruses, and we looked at antibody neutralization. We find that knockdown of target genes does not affect the antigenicity. Sorry about that. But so it's still not over because the genome has 19,000 plus genes. We identify, we screen, we validate, we come up with 124 hits, 60 of which validate between Hep2 cells and viral. Well, the question is, can dual gene knockdown further enhance the titers? So ideally, you want to knock down a gene that's in a different pathway than a gene you already knocked down. So here's your pathway analysis. It provides insights on how to validate your hits and integrate this into networks. So when you knock down different genes, different pathways, is this going to be negative, additive, or synergistic? So it turns out that dual gene knockdown is very useful. You can knock down more than one gene and get tremendous hits above and beyond what we expected. And here's just a couple examples. Well, we look at 54 gene silencing events with 7 different strains of polio. And we've got amazing results with dual gene knockdown. That's really important for the manufacturer because they all want their own tools. They don't want to use the same genes. Everyone wants to use different gene combinations, says their product's better. So we can sell that approach now, and we do. And you can see we're going from typically 20 or so fold increases. When you knock out this particular thing, you're now up to, what is it, about 100, 50, 60, whatever. It's just pretty amazing. But to tell you the truth, not all genes are good. You knock down genes from different pathways, you get 3 results. Generally, nothing happens. Doesn't matter. But the next thing that's most common is that cell line dies. They need those genes to grow. So that's a problem. And the third event that happens is this, where you see this enhancement. So just to give you some flavor of what this whole thing involves, the starting point is all these 19,000 plus genes in the human genome or viral genome. We're making flu vaccine, and they use MDCK cells, which were dog cell line. These numbers change, but it's roughly about 19,000. After a screen, we said, well, geez, we saw 124 events, which likely you knock them down, you get more virus back. We do a validation step where we deconvolute that pool of fluorescent RNAs to find out which ones are doing the job. We reduce that to 60. And as we look at live virus testing in viral cells, so this is all to date done in human cells, then we move to viral, which is the vaccine cell line. 50% of their hits drop off. So that tells you that monkey cells and human cells aren't even close. Everyone's, oh yeah, they're exactly the same. You can expect the same. It's all crap. We can tell you that's not the case at all. And this number was not expected, but about half of our hits don't work. Then you look at antigenicity testing, nothing drops off. So it's not the process. The way we would create more virus. It doesn't affect the vaccine. We tested for multiple strains. About 12 were selected because some of the ones didn't really work great in all the different polio strains. And then we went to industry with 6 or 7 hits. And they came back to us and said, single gene knockouts are great, but there's 20 companies that want this technology. You say, okay, no problem, we can do dual knockouts. So now we've expanded the portfolio that way. At the end of the day, though, we have to come up with a stable cell line, and we use CRISPR-Cas. This technology is pretty much in vogue right now. People will tell you, and incorrectly tell you, that you can use CRISPR-Cas plasmids to screen and knock out genes at the same time. That's okay if you're using human cells. Well, what if you're using dog or monkey or hamster? The cell lines that are used today in industry, those don't actually exist. So you have to use siRNAs, like I described. So the ease of the design implementation is the reason we chose the CRISPR-Cas editing system. It's very specific. It's actually very good. We've had mixed results at knocking out some genes, others quite easy. But the one thing you need to think about is knockout is not the same as knockdown. You can knock down a virus, a gene, and increase the virus. The cell line won't die. But knockdown, you see, like I told you, you only get about 85%. So you see a gene dosage effect where you have, if you knock out all the genes, the cells don't grow as fast. And so that's another rate limiting effect. But we can test that ahead of time. It's not a problem moving forward. But you just got to be cognizant of it. So these are actually knockout cell lines now. We're looking at here is 3 different genes. This is real life data for a company. And this is the wild type cell line that you normally use to produce the vaccine in. And this is the fold change relative of Sabin 1, Sabin 2, Sabin 3. We've got to compare dilutions because this was not set up right for this slide picture, but this is what wild type Vero produces. You knock out this one gene, and this is what you get for Sabin 1. This is Sabin 2, and that's Sabin 3. It almost kills all the cells. So you're seeing unrealistic and godly amounts of virus being made. So we're able now, like I told you, to really change the way the paradigm for vaccines are made today. The way all these vaccines are switching this technology because it's making it cheaper, faster. But one cool thing I wanted to bring up is how we found essentially the golden egg. So through this, we did our screen in polio. There's a whole bunch of coronaviruses that belong to this genus. Foot and mouth, AV71. In China, for example, you see AV71 has replaced polio as the major cause of disease and death there. So whenever a virus vacates a niche, it's filled right away. There's no niche unfilled. Something else will fill it in. Well, we found hits for polio that can be applied for all the viruses in the coronavirus family. And what we did is taking the same top 10 hits here, applied it for UV71, and we're seeing increases of greater than 60-fold. 60-fold. So that's huge because that allows us now to attack that problem right away. We've looked at Hep A. We've done a whole range of other ones as well, and it's true. This is being looked at right now with Muriel. This is one example. So testing targets from the polio strain allowed us to go after a whole other range of viruses. We've expanded this to a whole bunch of other programs, like I noted early on, looking at influenza, rotavirus, measles, etc. And without getting into any more data about this whole thing, we found that using different viruses with different cell lines in different assays all lead to the same results. We can find the genes that enhance virus replication. So these are in making cell lines for these viruses. And then those genes down here, we can target for druggable targets. We actually now can do it for polio, for measles, for mumps. We've tried it for West Nile, dengue, Coxsackie virus. We got the whole shebang. And so we're at the point now where we have technology, we're selling it to all the people to try and lower the cost of vaccines. The goal is having global access, right? You want to be able to vaccinate all these kids in third world countries that don't get that. You know that a combination of screening and gene editing can be used to engineer cell lines. We're seeing increases as high as 88-fold. This is a big deal. We know this applies to many viruses, We're finding the Achilles heel of the genes that control virus growth. That's a big deal because it helps us not only here, but in understanding the biology of how these viruses grow and creating new antivirals. And lastly, the ability to look at multiple cell lines. We're looking at the vaccine cell lines that are used today in the industry. We're not changing the way they're grown. We're just giving them a better substrate to make cheaper vaccine. So we're lowering the cost or increasing capacity in vaccine manufacture. And this is the Gates slide I wanted to bring up. The whole thing with Gates, the only thing they care about is polio, polio, and polio, and then polio again. But we have grants with them for norovirus, calicivirus. So their big whole thing is they're so vested in making sure their mantra of eradicating polio is done. That's what their focus is. So we have to do that first. Once that opens the gate to everything else. So our innovation at this point, and it allows us clear line of sight for these viruses and vaccines that need to be targeted for third world manufacture. So we're developing new vaccines that weren't affordable. They weren't made because they weren't affordable. We're looking at vector control. Zika is a great example of a virus that needs that. Enhanced manufacturing platforms. So instead of taking up your whole manufacturing facility for one vaccine, we can do multiple vaccines at the same time and increase the process development with all these guys. And so the last slide is thank you slide. Waylon is a member of the lab and drives most of this work at the CDC. This is the polio guy, Steve Oberste, the measles guy, Paul Rota. This is two people in my company that were working on driving this information to the vaccine manufacturers. And this is Carl, Carl Kirkwood, who's actually now at Gates. He runs all the enteric viruses, so the calices, the rotas, and all that. And it turned out that we had worked with him way before he went to Gates. Now we have a direct line, and this helps a lot, him looking it up. So just want to thank you for your time.